Hi again. In this short lecture, let's talk about the MHC proteins and antigen presentation, and then a short piece on the complement system that doesn't really seem to fit in anywhere else, so we'll sort of tack it on at the end here. So it started with the first slide on the MHC. MHC stands for Major Histocompatibility Complex. MHC. It's also referred to as the human leukocyte antigen. It's an antigen on human leukocytes. So you can see it by either of those two abbreviations, HLA and MHC. The idea is, it does a couple of things. This is a series of proteins on the cell surface. So there's lots of different proteins on cell surfaces. This complex of proteins serve as identification markers, both the MHC1 and the MHC2, those two classes. So this is how the immune system identifies whether your cells are yours or if they're somebody else's. And it forms the basis of tissue rejection. But these proteins are genetically determined and they are unique per individual. So your MHC proteins on identifying your cells as yourself are different from those on anybody else, unless you have an identical twin, in which case they would be the same. But again, these are cells that are proteins rather, that allow your immune system to distinguish you versus somebody else. And the idea then is to kill the other cells that aren't part of you. And this is the basis of histocompatibility. And histo is referring to tissues and whether or not tissues are compatible. And the idea is what accounts for tissue rejection. So that's one function, is to serve as ID markers. ID cells. And the other function is to participate in antigen presentation, which we'll talk about in this lecture. So, AG presentation. Those two functions of the MHC molecules. So the next slide, I need to do some drawing. So, I skipped one. I'm going to start with MHC1 proteins. And MHC1 proteins are on the surface of all of your nucleated cells. So every cell in your body that has a nucleus, so we're primarily excluding red blood cells, pretty much all the rest of them that have a nucleus have MHC1 markers on them. Now remember, it serves as an ID tag, and it also is going to serve in the function of presenting antigens. So ID tag is one function of these MHC molecules. The other idea is with antigen presentation. And what we're going to talk about are endogenous antigens. So endo is from inside. So let's start with a cell. And this is one of your body cells that has a nucleus. They all do. And the idea is that if these cells, your body cells, become infected with a virus, how can we get rid of it? So let's pretend that we have a virus that gets into the cell. So we'll have a virus or a viral infection. So viruses, coronavirus, not any different than the rest of them, has to be replicated inside your cells. And viruses are specific for the cell type. With the coronavirus, COVID-19, its target cells are those of your respiratory system. It's like any other, it's like most other cold viruses, influenza viruses. They target the host cells of your respiratory airways. So at any rate, the virus gets inside, and then the host cell, your cell, needs to replicate the virus. So we wind up with this business of viral replication inside your cell. At this point, the immune system can't see what's going on inside the cell. So as the virus replicates itself, it makes viral proteins. Viral proteins are synthesized. Your cell is taken over by the virus, and it's your cell that makes more of these viral proteins. So let's pretend that this is a viral protein. I'm just drawing some sort of random structure. So this is a piece of the virus. So these proteins, they get made. And they will eventually get assembled into a virus. So your cell inside is making these. And then those will go on to assemble, and then they will release more virus out into the rest of the body to infect other cells. Well, this cell is diseased, and it's been taken over by a virus. And what happens is that your cells are constantly making these Mhc1 proteins. So this is normal protein synthesis inside your cells. And we make an MHC1. And I don't know how to draw an MHC1, so I'll do it something like this. And I'll put a little groove in it. And we'll just call this MHC1. It's a protein that gets stuck on the cell. And what happens is that these viral proteins, now these are endogenous proteins. These are inside. So let's write that on here also. The virus came from outside, but the proteins that are made are now inside the cell. So they're endogenous antigens. And we'll abbreviate antigen as AG. And those antigens inside the cell get attached to the MHC1 in that groove. So here is the little piece of the viral protein. The idea is that we're going to present this to the outside world. That outside being everything outside of the cell. So they get together, and then this gets exocytosed onto the surface. And we have something that looks kind of like this. Oops. Need to make a little groove. Clean up the inside of the cell. And there's our groove. And the protein, the viral protein that was inside the cell is now stuck to the MHC1 on the outside, and we're sort of waving it around, if you will, to the rest of the immune system. Say, hey, come check this out. So that's our MHC1. So this is antigen presentation. Antigen presentation of an endogenous antigen. And then what happens, excuse me, are other cells of the immune system, T helper and cytotoxic T cells come by. So antigen presentation, let's say that it is presented to T helper cells, so I'll just abbreviate T sub H, and cytotoxic T cells that are wandering around, and they bump into the MHC1. They're sort of this secret handshake. They recognize MHC1 as being normal body cell, but then they also recognize this foreign antigen, and they say that's not supposed to be there. So then the cytotoxic T cell kills the host cell. So it kills, it destroys, it lyses the infected body cell. Yes, we kill the cell, but we also kill the viral replication factory going on inside it. So the cell is killed, and we start to eliminate virus production inside the body. So again, this is MHC1 story and the presentation of endogenous antigens, those antigens coming from inside the cell. Now this normally occurs all the time for other proteins that might happen to be inside the cell, such as bits of worn out mitochondria or worn out organelles of other type that are inside the cell. They get damaged, destroyed. The MHC1 is constantly putting stuff on the outside and sort of circulating what's going on inside to the outside, so other cells of the immune system can monitor. And if it's a normal piece of mitochondria on the MHC1, no problem, leave it alone. It's recognized as being part of yourself. But if it's not recognized, the antigen is not recognized, then the rest of the immune system kicks in and we remove that cell from the body. That's the basic story. The next slide is with MHC2 and exogenous antigens. So on this story, we're dealing with MHC2, and these are on APCs, or antigen presenting cells. I'll abbreviate antigen as AG presenting cell, or cells. And these are typically macrophages and B cells. Okay, so we are dealing with exogenous antigens. Antigens that are coming from outside the cell. So let's draw another picture of a cell. Something like that. So this is our APC, our antigen presenting cell, either a macrophage or a B cell typically. And here, so these are phagocytic cells, for example, the macrophage, and here is our bad guy. Let's pretend this is a bacterium, and it has different things. I'm just trying to draw little proteins on the surface. It doesn't really matter what it is, what it looks like, but let's label it as a bacterium. Or several bacterial cells. And being a macrophage, it endocytoses it. So I should have drawn my membrane a little differently here to start. That's why you're doing yours in pencil, I hope. And, oops, we want to show endocytosis here. So here is the bacteria again that's being endocytosed. You know, this basic story. Endocytosis, that then enters the cell, and we have a phagosome that has the bacterium inside of it. And you know what we do? We digest it. So we bring in another organelle, a lysosome. This goes back to Biology 160. And you remember that lysosomes have digestive enzymes inside them, proteases, hydrolytic enzymes. And these two merge. And we have two membranes together. We have a phagolysosome, if you will, and the bacteria gets broken down. And so here are little pieces of the cell wall, fragments. And then we have some, digestive enzymes. And so what happens here, so we've endocytosed this bacteria from outside. So this came from outside, so it's exogenous. Exo from outside. And again, we have protein synthesis going on inside the cell. I'll abbreviate synthesis, protein syn. And we are producing MHC2 molecules in this case. And so here is our groove again. So this now is an MHC2 dealing with exogenous antigens. And what we have is that those fragments then get stuck in the groove, and there's a piece of that original bacterial membrane with the antigen that's stuck to it. So this was our bacterial antigen fragment after digestion. I'm abbreviating fragment. And then this gets exocytosed to the membrane also. So again, this is our MHC2 functioning as an antigen presenting protein and our antigen fragment. And so what we're doing now is waving this fragment, presenting this antigenic fragment to the rest of the immune system. And so in this case, this antigen presentation sort of sounds or raises the alarm. So antigen presentation let's say raises the alarm or sounds the alarm. And this is involved with activation of T cells and helper cells. So let's just say activation of other immune cells. Immune system cells. For example, again, T helper cells and cytotoxic T cells. There are chemical interactions that occur for the activation of the other immune system cells. But the rest of the helper cells come along and recognize that antigen fragment as being foreign. So what do you want to do now to the antigen presenting cell? We don't want to kill it because there's nothing wrong with the cell itself. It did its job. It destroyed a bacteria and presented fragments of it to the rest of the immune system. So don't kill the messenger, if you will. So do not kill cell. Give it a medal or a reward for identifying the bacteria as foreign and raising the alarm. So antigen presentation, MHC1 molecules for endogenous antigens, MHC2 for exogenous. And again, MHC molecules also serve as identification tags. Okay, the last piece that I want to go over is just to introduce you to the complement system. And as I mentioned before, it doesn't seem to really fit any other place. It's somewhat independent, although it's all working together. It's been studied, I guess, independently as part of a complicated system. So it got its name originally because it complements other immune system functions. So that makes sense. So we'll just call it a complement system. It consists of numerous plasma proteins. It's a rather long list of them, and they are made in the liver. And when activated, this system enhances or complements both innate, which is non-specific, and specific immune functions. So it essentially activates or complements all of the immune system functions. It involves a cascade of reactions, which is where some of the complication comes in, and different pieces, different fragments have different functions, and it's we're not going to go down that path. One of the things that happens, sort of the big picture, if you will, is that these complement proteins, these complement fragments, they wind up binding to cell membranes of pathogens which label it for destruction. Phagocytosis. So that's one of the functions of the complement system, is to tag antigens with labels that say, destroy me. You're painting a bullseye on the pathogen, in a sense, for other cells of the immune system. The complement system also serves as a chemotactic agent. Chemotaxis is just movement toward certain chemicals, and that attracts different phagocytes, so that's another function. And the complement system also forms what's referred to as a membrane attack complex, which is inserting holes in the cell membrane. And then that leads to lysis, destruction of the cell. So those are three major functions of the complement system. Enhances phagocytosis by opsonization. We'll talk about this again when we talk about antibodies. Look it up if you're not familiar with the term. The complement system acts as a chemotaxis agent to attract other phagocytes, and it punches holes in membranes. There's a little bit in the book on that, but I would not get bogged down on it. If you remember these major functions of the complement system, and it's a series of proteins made in the liver that, when activated, enhance both innate and specific immunity, I think you're good to go. So, great. Next we will talk about B cell and T cell activation, part of specific immunity. I hope this helps. Ask questions when you have them. See you next time.